Hello everybody. I just met Simon last month and now we are friends. So I come from the Queensland Institute of Medical Research and you can see the Berghofer there. I'll tell you a short story. He donated 60 million to my institute. And that's why we have changed our name now to his surname to QIMR Berghofer. So everything will be changed from our calling cards, from our ID, from our lab coats, everything. But it's good to work in this institute. Come over at Brisbane and I'll show you. Okay, what I'm going to share with you today are all results of unpublished work. So we haven't published this yet, but they're all exciting and promising, and hopefully we can make good use of my diagnostic test. And of course, you see all those people there at the institutes. It's a big collaborative project, but it's only a small pilot project funded by HotNorth. And HotNorth is a group, it's a program grant by the National Health Medical Research Council of Australia, and it's all promoting the health of people that live on the tip of Australia, and it's hot there, so that's why it's called HotNorth. As we all know, diagnosis of scabies is challenging, especially the early stages of the disease, because it just mimics other skin conditions such as eczema, psoriasis, dermatitis. And presumptive clinical diagnosis is basically just based on nocturnal pruritus, location of papules, or history of exposure with infected individuals. It's only recently that we have this diagnosis published, like based on the Delphi method, and we've heard Daniel talked about them. And also, the definitive diagnosis, as you would see there, is microscopic confirmation of standard skin scrapings, which often fails because these mites are elusive. You know, ordinary scabies will only have 1 to 5, and when you scrape them off, they're gone. So you won't see them, guaranteed. So why are we concerned? Well, of course, misdiagnosis have a domino effect. It leads to improper treatment, which improper treatment leads to serious disease sequelae, and of course, increased chances of epidemics. And further increase in misdiagnosis will also result in inaccurate assessment of the effects of your intervention. And of course, you will say that my control program failed. So it all starts from misdiagnosis. And what are our alternative tests? Of course, we have dermatoscopy. We've heard about these video dermatoscopes, which can increase resolution so you can observe directly the mites on the skin of your patients. And these are very high resolution reflectance confocal microscopy and optical coherence tomography. These are this one is you can just look at the skin and it goes from epidermis to the dermis. So it's non-invasive yet. These are so expensive, and of course they're used in high-end dermatology labs. And any high-technology gadget, if it goes to use by inexperienced people, will still lead to misdiagnosis. In Darwin, we have heard how the nurses experimented on dermatoscopy, and the images that they just get are not really clear. You really need a lot of practice and a lot of experience to really make all these high-tech gadgets work. But definitely, they are non-invasive. If you get to use them, you don't need to scrape the skin. You don't need invasive techniques, so you can visualize the mite. Because it's actually seeing the mite that will tell you that it's really scabies. Of course, in the laboratory, or you can also do the Barrow ink test. You just follow this. But as earlier said earlier in the symposium, this is rarely seen. And also, you can look at mite. You develop antibodies against the mites, but there are many publications looking at your antibodies against scabies mites that they are really cross-reactive with house dust mites, so they're not really helpful. And of course, our very favorite PCR of which there are several targets. You know, it's a DNA test, you amplify, you make many copies of your DNA from the scabies mite, and these are the different targets, ribosomal DNA or your RNA or mitochondrial DNA, all of which have their own merits, but let me take you to our project. We have three aims for this. We would like to develop a highly sensitive and specific diagnostic probe-based quantitative PCR, and as you've heard earlier, quantitative PCR does not look anymore at gels, so you quantitate the copies of your target and all you see are all these graphs. So we want to make this, but the difference is we want to look at the target of the scabies genome that is more abundant. And if it's more abundant in the scabies genome, it means that we can increase our sensitivity over and above the existing PCR assays. And we also wanted to look at a very optimal genomic DNA extraction technique for the mite samples, because it's ensuring maximum yield for molecular assay. Because your PCR may not work, it's because you were not able to extract the DNA properly or you didn't have enough. So it's like looking for a best DNA extraction protocol so that I can tell you that my PCR really works because I have an optimal DNA extraction technique. And the third objective is, of course, everybody shies away from skin scrapes. We wanted to look for a very effective non-invasive method as an alternative of sample collection, and we look at swabs here. We tested two kinds of swabs so that we want to ease the diagnostic workflow. Skin scraping using a scalpel blade is of course invasive. How did we go about this? Because the scabies genome is now published, there are many, you know, the whole scabies mite genome from the pigs or human scabies mite, they're all published in the literature. So we mined for looked for the most abundant DNA sequence in the genome. This is so that our assay will be more sensitive. And out of that, we identified two targets that we tested in this project, and that's where we designed our primers and probes. And with the help of a bioinformatician, there were 6 targets that was given to me, and I singled out, you know, these are just the 6 targets that he identified that I can design my primers and probe for the PCR. So I singled out 2 that I will test. And I also looked at conserved regions. Conserved means that the sequence of the pig mites and the human mites are the same in the genome, and that's where I chose my targets. And also there are unpublished scabies genomes, and they're hiding from the repository of WIHI, so they gave us access to their mite genome sequences. So because I need to confirm the targets that were identified by my bioinformatician and the 2 targets of my PCR that I have developed the assay. So we would like to thank the WIHI collaborators for making this unpublished genome sequence available to us. So once we are sure that our target exists, because you know, next generation sequencing technology, they have many technologies to make the genome, but this one is so far gives very long repeats, very long regions. So I think this is the most reliable at the moment, the PacBio generated sequences. And we also looked at different mite disruption methods because, you know, it's a tiny speck of dust, but you really need to crush the exoskeleton because it's quite hard. So it doesn't mean it's tiny that you can immediately or really do extracting DNA straight away. Even if it's tiny, no, it is a hard exoskeleton. It's an arachnid mite. And we also experimented on different extraction protocols. There are kits, you know, there are easy ways to do it, to find out which one will make our PCR work better. And this is what Simon is saying, we had the luxury of availability of scabies mites. So we have developed an animal model at the University of Queensland. This is over 100 kilometers from where the QIMR is. So what we did was to have colony piglets, and we can grow scabies in their ears. And how do you know how we make them susceptible to scabies mites? We treat them with steroid. As you know, encrusted scabies, if you are immunocompromised, you're a good host to scabies mites. And well, we started from injecting steroids once a week, but because the steroid is, injection is invasive, you know, we have trained our pigs to swallow steroid tablets with marshmallows. And these are our marshmallow pigs, we call them, and they're very susceptible to scabies. And we just confine the scabies in the ears, actually. You're not, you know, animal ethics of you will say, don't, you're not going to infest the whole body or you'll be in big trouble. So this is our resource of available mites. That's why I told you that I have designed my primers and probes on the region where it's the same as the human mite and scabies mite. Because, you know, there's not a lot of scabies mites, I already said, unless you find a really crusty patient. So this is our resource. So we optimized the assay using mites from our pigs. We checked our swabs by swabbing our pigs. So all the assay optimization, we used our animal model of scabies. And of course, we check the performance of our optimized qPCR assay by asking our collaborators from Darwin, from the Royal Darwin Hospital, to collect samples from patients admitted to Royal Darwin Hospital, and Darwin Dermatology. Darwin Dermatology, we have more cases of other skin conditions because it's a private dermatology lab, and these, whereas here, these are hospital admissions. Here are some results. So we've compared other methods of homogenization using vessel, beads, tissue homogenizer, and combined it with different kits. As you can see that you increase the number of DNA copies here of the different targets. You know, I call my targets SSR5 or SSR6. That stand for Sarcoptes scabiei region 5, Sarcoptes scabiei region 6. And our reference assay here is COX-1. So if you're developing a new test, you must compare it with an already published assay, so you know how you fare. So these are just results, and you can see that you increase the recovery of the DNA when you subject your mites with homogenization. And of course, we have to look at assay sensitivity. So the two targets, we need to clone it in a plasmid, and you make a standard curve. So CQ means the lower limit of detection you have, or these numbers here, the better your diagnostic assay is. So as you serially dilute your plasmid DNA, this can give you how many copies you have in your PCR reaction. And as you can see here, the limit of detection for both assays, SSR5 and SSR6, is 30 copies of the targets per 10 microliter reaction. So this is how sensitive the new PCR assay is. I'm talking about SSR5 targets in 10 microliter reaction, so 30 copies. But we wanted to translate it to how many mites can our assay detect. So we put here on the left panel 1 mite, 2 mites, 3 mites, 4 mites, 5 mites. And you can see the limit of detection here is lower for one target, which is SSR5 and SSR6. So it means if I compare the performance of my two targets, one stands out better than the second because it has lower CQ values, or your threshold of your quantitation here. And you see, to look at this, if the differences in the performance of the targets are significantly different, of course we did some statistical tests, and as you can see there, my SSR5 works better than my SSR6. Look at the p-values there. They are different, but really the two targets are performing differently. And if you compare it with the published assay, my SSR5 works better with lower CQ values here than the SSR6. So it means that my new assay performs better than the published assay, but only one of my candidate assay, which is SSR5. And of course, we subjected this again to the statistical test, and this difference here is significant. So specificity. Well, we need to check if our PCR is going to detect other mite species. So we checked it against house dust mites, or this Dermatophagoides farinae. Or there is an eyelash mite in humans, but we can only get the canine one, which is Demodex. We've tested it against head lice. We've tested it against common skin pathogens, Staph aureus, Strep pyogenes, P. acnes, P. rubrum, and T. interdigitale. So in all of this that we tested, we never got any amplification, meaning that the new PCR test is specific. And we also checked human skin and pig skin so that we make sure that we're not amplifying or PCRing host skin, and it's all negative. And also, we also tested our targets from DNA extracted from our patients coming from Darwin Dermatology, and they have dermatitis, eczema, they have psoriasis, they have tinea, they have pityriasis versicolor, they have seborrheic keratosis, and they're all negative. And this is for Simon. This is your standard PCR gel. Simon told me that ages ago. These are his PCRs, so here you go. See, that's the panel there, SSR5. There's no band. You know, if you see a band there, then your PCR is positive. So we didn't detect any in the human skin on this lane, pig skin on this lane, Demodex canis, the house dust mites that we serially diluted. And these are the other skin pathogens, the bacterial ones, the fungal ones. See, no bands, so they are negative. Oh, I forgot to tell you about this one. Oh, as regard the sensitivity, this single mite, we put it also in one tube, then we call it neat. And then that single mite, we extracted DNA, but we diluted it tenfold. So it means this one here, that point there that got amplified by our PCR, that is 0.1/10 of a mite, because it's 1:10 dilution. This is 0.01 or 100 of a mite, and we were still able to detect it. So it means our PCR assay can detect single mite, 1/10 of a mite, or 100 of a mite. So that's how sensitive it is. And of course, we've tested two kinds of swabs, and one performs better than the other. This is the flux swab in terms of DNA copies there. See those blue? They're much higher than the DNA copies detected by the catch-all swab. And of course, if you look at the CQ values, you can see here the ones in blue are your flux swab, and check if they're significantly different from each other, and of course they were. So one type of swab outperformed the other one. And these are the panel of our other skin conditions collected from Darwin Dermatology. These are the clinical diagnoses and the site of collection and our qPCR results. We say it's negative when it's been tested with target 5, target 6, and the reference target, so highly specific because we didn't get any PCR. They're all negative with other skin conditions. And here is our magic slide for clinical samples. I'll take you a bit here because this is really interesting result. So for those, well, these are patients admitted at Royal Darwin Hospital. This is the clinician's diagnosis, and as you see, I have a panel of ordinary scabies. These are the early stages of the disease, not the crusted ones. And these are where the samples were collected. And I've also asked them to look at the history if they've been treated. So I'll take you first to the ones that treatment have been given prior to admission. This one, that one, that one, and that one. Our PCR were all negative in those three instances where treatment was administered. Or 4, they're all negative. And I think there was one question that Simon asked me, how long after your PCR will work? Well, this one, I checked the case report forms, and it's a week after treatment. They are negative already. This is 1 week prior to admission. And we also, because there were also skin scrapes that I asked the clinicians to collect for us. And once they've collected, they take it to Menzies School of Health Research in Darwin. They look at it in the microscope. And these are the microscopy findings. As you can see here with treatment, microscopy and PCR agrees. Here and here. But here, look, microscopy, there's 2 question mark here. One's treated and one's not. But I guess this one is they didn't really see the mites because I have notes also in the CRFs. They said embedded in the skin. And because they didn't do the KOH mount, you need to tease the skin to really look at the mite. And what they said is, yeah, we assume there's scabies there because of course they're relying on our magic clinicians here because they're admitted they have ordinary scabies. So they are embedded, but they didn't really see it. So that's why in the result form, I had a question mark there. But look, that was collected on the right foot, this is on the left foot, and it's negative here. So what can we deduce from this diagnosis here? Is this really scabies or is this a clinical loophole in the diagnosis? See? And also, the very good asset of the PCR is, of course, when microscopy is negative, in this case, the green ones, qPCR is positive. 1, 2, negative with microscopy, qPCR is positive. Negative and positive. But of course, I can't rule out a false negative reaction, if I should say, in the point of view of PCR. But of course, if that microscopy is questionable, then most likely this is correct, but we don't know. See, though, I'm just showing you that clinical diagnosis of scabies can be really that challenging, so it's for us to deduce if we really trust 100% the clinical diagnosis. But if we follow those Delphi classification, and next year I think there's going to be the IACS clinical diagnosis, if you pair it up with qPCR, then you may be able to nail the diagnosis, Simon. So in summary, we have developed a simple DNA extraction method that has been optimized to ensure maximum yield suitable for this molecular assay, and we have found a non-invasive sample collection tool, which is the use of swabs, which will definitely ease diagnostic workflow. And we can detect 1 mite with this PCR, or even a fraction of a mite, this 0.1 or 0.01 mite material, because essentially for a DNA test to work, you only need 1 molecule. And it's going to work if it's highly specific and highly sensitive. And of course, we have a highly specific test looking at the panel of the other skin conditions. We've tested it against bacteria, we've tested them against other species of mites and other fungal pathogens in the skin. They were all negative. And might new target, because it occurs in the highly abundant region of the newly published scabies genome, that makes it performs better than the published assay. In the future, well, at the end of the year, I'm taking these swabs to the Kimberley communities. You've seen those Teleton kids. They have a big survey of school children, and they do surveys on skin pathogens, and scabies is included. They're also swabbing everybody. It doesn't hurt if I ask my collaborator to just 1 swab for me, so we can put this assay to test, isn't it? More testing, more confidence that you can use it in a large scale basis. And of course, this is a work of so many collaborators from QIMR, James Cook University, Walter and Eliza Hall, Menzies School of Earth Research, Royal Darwin Hospital, and this is my Western Australia Kimberley collaborators can attest this in the last quarter of the year. And this is where we keep our animal model at QASP in Gatton. And this is just a little pilot project funded by Hot North, not the millions that Daniel had. But we hope to make a difference. Thank you.